Hello, I'm Aurélien Roux.
And I'm Sandrine Morlot.
We are both from University of Geneva and we would like to present you the data we are publishing in this issue of Cell. In clathrin-mediated endocytosis, separation of the clathrin-coated pits from the plasma membrane is mediated by dynamin. Dynamin forms helical collar around the neck and constricts upon GTP hydrolysis to promote membrane fission. However, this constriction is not sufficient for membrane fission. Elasticity of the membrane is thought to play a role in fission, as actin and longitudinal tension along the neck are required for fission. In this article, we studied the role of membrane elasticity in controlling the location and kinetics of dynamin-mediated membrane fission.
Using this system, we first observed that fission occurs at the edge of the dynamin coat. As seen on this movie, the break occurs just at the border between the green dynamin seed and the red membrane tube. In collaboration with Martin Lenz and Louis Diniz, we hypothesized that the elastic stress of the membrane at the edge of the dynamin helix could favor fission by reducing its energy barrier. In this case, theory predicts that the kinetic of fission depends on the elastic parameters of the membrane. As shown on these graphs, the average fission time increases with increasing bending rigidities and when lowering the membrane tension.
However, to further confirm our model, we ought to evaluate the mechanical work spent by dynamin in constriction. This would allow us to verify that dynamin is strong enough to constrict a membrane and that the energy spent by dynamin in constriction is enough to reduce the energy barrier to fission.
Dynamin constriction is partially mediated by a torsion of the helix. This torsion is generated by the application of a torque. A torque is a rotational force multiplied by the length of the lever arm. Our collaborators, Marius Klein and Giovanni Capello from the Curie Institute in Paris, measured the maximum torque delivered by the dynamin. To do so, using magnetic tweezers, they block the rotation of magnetic beads attached to constricted dynamin tubes. As seen on this graph, beads stop rotating for values close to 1 nanonewton/nanometer.
Our results show that dynamin generates enough elastic stress in the membrane to trigger spontaneous fission at the edge of its helix. Our study explains why dynamin constriction is required but not sufficient for fission. Precise control of membrane elastic parameters is also required. Our results are consistent with previous findings showing the requirement of longitudinal tension and attachment to the substrate in fission. It is also consistent with actin polymerization as seen in vivo. From this study, we can further envision the exact sequence of events in dynamin-mediated membrane fission as seen from this animation.
In endocytosis, dynamin dimers associate in a helical collar at the neck of the clathrin-coated pits. Upon GTP hydrolysis, dynamin undergoes a conformational change leading to torsion and constriction of the helix. Fission happens at the edge of the dynamin coat where membrane stress is the largest. The protein components disassemble to be recycled.